Welcome to Mr. Chalk's Revision Tips.
When answering exam questions, there are a few key things to remember, right? So just remember, exam technique is a key part in preparation for any exams, and it's something that you'll pick up the more you go and you do past papers. Right, so many questions in exams are repeats of what will be in old exams. So going and looking at lots of generic questions beforehand is a good idea. When going and answering and questions and reading them, there's a few things just take into account. Make sure you go read the question at least twice so you don't make any mistakes when you're reading them. As you're reading them, go highlight key information. Don't make assumptions about what the question is asking. Make sure after you've answered it, you go check your work for daft errors that you might make. And if it involves going and answering something where there's some calculation, make sure that you write your workings out down. Right, let's go and have a look at the question.
The Operon consists of 3 different structural genes, so a promoter, a terminator, a regulator and a operator. So the 3 structural genes are for LAC-Z, LAC-Y and LAC-A. LAC-Z encoding for beta-galactosidase, LAC-Z going and coding for some sort of intracellular enzyme, and all of these things are there to allow the bacteria to go and metabolize lactose. So an operon in general is a group of genes which will go and code for a specific task to be carried out by the bacteria. So the genes will go and produce proteins which enable the bacteria to break down lactose, which they only need when lots of lactose is present and no glucose is present. Normally a repressor protein is bound between the promoter and the operator when there is no lactose present, preventing the transcription of these 3 genes. So like we said, bacteria usually goes and respires or metabolizes glucose. However, under certain circumstances, it will be able to use galactose as a respiratory substance that it is producing by the breakdown of lactose into glucose and galactose. So if we just go and think about how they can go and use this galactose. So it's things like the fact that the bonds in galactose also contain energy that the bacteria can use for respiration. Galactose is soluble, a bit like glucose, so it can move within the cell and can form hydrogen bonds with water, which also goes and increases its solubility. Right, if we go and look at a question, so one of the monomers of lactose is galactose, like we just looked at. So bacterium E. coli usually uses glucose as a respiratory substrate. Under certain circumstances, E. coli is able to use galactose as a respiratory substrate by breaking down the lactose into glucose and galactose and then respiring both. So we're going to go and explain how the structure of galactose allows it to be used as a respiratory substrate. Same again, like we just looked at, talk about the fact that the bonds contain energy, and that energy can be released by respiratory enzymes. Talk about the fact that because you've got that H and an OH group in it, that will allow it to form hydrogen bonds with water, which goes and makes it soluble. If we go and look at this second part, so E. coli usually grows in conditions where the extracellular concentration of lactose is relatively low. So in such conditions, lactose does not easily cross the bacterial cell membrane. Right, suggest and explain why lactose is unable to cross the membrane. So that's just basically talking about maybe the size of the lactose. So you need to talk about the fact that as a molecule it's too big, so it can't go and pass between the phospholipids or through that phospholipid bilayer. So the fact that the concentration gradient remains small, and you'll need some sort of carrier protein or protein pump in order to go and bring that lactose in. So hopefully you have found that useful. If you've got any questions, then drop it in the comments below, and we'll be uploading a lot more. Right, good luck, folks.